Okay. We begin. I'm gonna teach you about these. Woo! Yay! All right. So anyway, I always have to start at the videos like that.
You sound so excited.
To be fair, this stuff is actually super cool. Okay. So.
Then act like it.
Gross. Muscles. Yeah! Like that.
All right.
I'll take it. Okay. So. Pretend that's a muscle. Ooh, wow. And if I do a cross section, that turns into this, and then it's got a bunch of basically like muscle fiber bundles. And then if I take a cross section of, if I do that, boop, go there, pull out that, it's got a whole bunch of little of these ones as well. Yeah, that's good enough. And then if I take one of those, one of these, that, just a single one of those is a muscle fiber. So I'm going to draw it out that way. And so they're repeating units of stuff called sarcomeres. So this whole area, that is one sarcomere.
Mm-hmm.
It's a really, really basic, simple anatomy of that stuff. So I'm going to erase the rest of this because it doesn't matter anymore. And I'm going to draw a very, very large balloon of sarcomere. So top and bottom lines, that's just the edges of the muscle fiber. Okay, you've got that. And then you've got these. Yeah, we'll draw these and that. So you've got this is my super anatomically correct drawing of a sarcomere. Okay.
Or a candy wrapper filled with fuzzy caterpillars.
Yep, fuzzy caterpillars. I'm going to draw. Alright, I started drawing out circles, but I gave up. Okay, so these, the zigzags, that's what's called the Z-disc. The disc. And this is the base of attachment for these strands. So the small ones, those are called thin filaments, and then these are called thick filaments. Your thick filaments is where your myosin heads are, and that's what does all the contraction in your muscles. These, so the thin filaments are made up of the dark blue line, which is a nebulin protein, and then the blue is an actin helix, double helix that wraps around it.
Mm-hmm.
And then I will blow that up. So here's our nebulin filament. And I will draw our actin. I'm so good at drawing circles.
Oh, yeah.
And I'll do the other one. So anyway, there's that. Actually, that's not too bad.
Mm-hmm.
So there's the double helix, and then sitting on top of these actin. So each subunit, so just the little one, that's called alpha subunit for actin,
Yep.
and then once they're all together, it's called F-actin, hence which stands for filamentous actin. And then running along top of that, there's another double helix protein, and that one, I'm going to draw that one in green. So actin, fun little ball, and then right on top of it, here's its little binding site. And so this, the red and green, so that's what's called tropomyosin, and it sits right on top of the actin, and which will make sense later for once I start talking about myosin. And then there are, I've got all that stuff, we'll use black. And then sitting on top of that, we've got what's called a little Mickey Mouse. Kind of hilarious, but I like it. So tropomyosin, actin, nebulin, and then that was called troponin.
Mm-hmm.
And so it's got 3 subunits. It's got troponin I, T, and C. The troponin I binds to the actin. The troponin T binds to the tropomyosin, and the troponin C will bind to calcium, which is what causes basically all contraction within your muscles. Any questions?
Nope.
So that's the thin filament. Thick filament, so we've got the base. It's like a whole bunch of tails, but I'm just going to draw one. And then, so this is the myosin head, neck, and then the tail.
Mm-hmm.
And each myosin consists of 2 heavy chains and then 4 light chains. So really, myosin head is like that. It's got 2. So 2 heavy chains and then 2 light chains somewhere there. We're not really sure where, so we wave our hands. Not even kidding. They're somewhere on there.
Yep.
And then at rest, the myosin head has ADP bound to it and then a phosphate group right there. And so this is what's called the cocked position. Okay, active myosin, that describes all of that stuff. Yeah, so Z disc, and then thin filament, thick filament, so on. And then this is just repeating structure throughout all muscle fibers, and then all your muscle fiber bundles, and then the muscle belly. Good? Good? Great.
Do you get to memorize the whole cycle of the
Yeah, I'm gonna talk about that.
Okay.
So that's gonna be, we'll talk about that actually once I'm done talking about action potentials and such. Okay, so I'm going to draw again this. So there's I want this color. So here's our neuron. This is our alpha motor.
Beautiful neuron.
There's our alpha motor neuron. Action potential comes down. It's got a bunch of vessels that have acetylcholine. Acetylcholine goes in here, opens up fun little channels that are ligand gated, that then open sodium to rush in, which then, finish drawing this. This is a really cool thing. And then I'm going to draw this. Pretend that's something interesting. It is actually. So we've got that one, and then we use that color. So then we'll use this one. Okay, so what happens? Sodium comes in, starts depolarizing the membrane of the muscle fiber, and then this right here, it starts opening voltage-gated calcium channels. As
what's the red cell? Is that
the red? So that's the muscle fiber.
okay? So once that starts,
So this starts depolarizing. So
right,
we start
because of
accumulating
sodium influx,
that
and then it opens the
calcium runs in,
Voltage-gated.
and then same thing down here and down here. This little channel, this is what's called a T-tubule.
Mm-hmm.
And so this is extracellular fluid. It has a lot of calcium in here, and so as this rushes in, depolarizes, this is what's called a DHP channel,
Mm-hmm.
and so it's basically just a voltage sensor for, this is all just skeletal muscle. And so the way it works, it's really interesting. It's basically directly connected to this channel, which is called it's a ryanodine channel. So there's a high amount of calcium in this. So this is what's called your sarcoplasmic reticulum. It's basically your rough ER, but within muscle fibers.
Yeah.
So
Yep.
super high in calcium, and it's like a huge, like plexus all
Yeah.
over muscle fibers.
Doesn't look like that. I understand.
Yeah, this is for my simple drawing.
Not a potato in a cell. Got it.
No. So when the DHP senses, when it changes based off of the voltage and this becoming depolarized, it opens, but as it opens, it also opens the ryanodine channel. So it's
Interesting.
kind of like a lock like this. It opens and pushes the other one open. So that then allows the calcium to rush out, flood the cell even more, which then causes the muscle to contract. And the interesting thing is the driving force for calcium to leave the SR as opposed to coming in from here, it's way stronger for the calcium inside this to come out. So basically none of the calcium outside the cell actually goes in because of this channel opening.
Mm-hmm.
Hence why we like to refer to this one basically more or less as just the voltage sensor. Yeah. So calcium controls basically all contraction. So with troponin, so you've got 4 calcium binding sites, those 4 little lines. So this is the C subunit. So it already has calcium on 2 of them, that's calcium. When the other 2 bind to the actin, it takes the actin and the troponin and moves the tropomyosin, which then allows the myosin head to bind to the actin binding site.
So the protein has a conformation change and pivots the.
Yeah, once the 2 other calciums bind to it.
Okay.
Hence why, literally for anything with muscles, it's all dependent on calcium.
Mhm.
So once you get this flood of calcium into here, you start contracting our thin and thick filaments.
Right.
And so there's all that. Make sense? Great.
All good, all good.
Okay, now I'm going to draw a fun diagram. Okay. It's not that way, it's this way. That's our phosphate. So Z-disc, thin filament, thick filament, here's our myosin head. So this is in our, what's called the cocked position. It's ready to bind, but the tropomyosin is blocking the actin from binding. And then what happens, we go to the next stage because we have a huge influx of calcium. And so what that causes is the myosin head to bind to the actin. And then as that binds, that causes the
Phosphate.
the phosphate to go away.
Mm-hmm.
And then the next step that's what happens.
Much
So-
better. I like that.
Binds there. Phosphate is still on. And then at this point, then the phosphate comes off.
And then it fires.
And then this is what's called the power stroke.
Mm-hmm.
So the phosphate comes off as the myosin head binds to the actin and then pulls in.
Yep.
So then we've got that, and then there's no more phosphate. And then the next thing is we have ATP that comes along in here. And when the ATP binds to the myosin head, it binds and it pulls off. I'm going to draw our little, that's the ATP molecule.
Mhm.
It causes it to unattach, detach from the actin. And then moves here, hydrolysis. So basically the entire time we have an ADP on there.
Yep.
The ADP comes off here as well. And ADP. Just assume that there's an ADP there. So well, after the ATP binds to the myosin head, the myosin head has a hydrolysis enzyme with it. So then that breaks the ATP into the phosphate and ADP, recocking it. So cocked binds, this is now uncocked because it's contracted in, and then this is the recocking, and cocked again.
Yep.
And that's the general structure for skeletal muscle. And skeletal muscle uses basically 100% of the sarcoplasmic calcium. It doesn't use anything from the extracellular stuff.
Right.
Cardiac muscle, because you want that to continue beating, is 90% sarcoplasmic stuff and then 10% extracellular. And then smooth muscle is about a 50-50 because you have some muscles like one of the best ones in class is like your internal anal sphincter. You don't want that loosening.
It's true.
So because you can use the extracellular stuff, it can just stay contracted for a long time. And the difference between that is, so for skeletal, you had the DHP and the ryanodine like basically linked. In cardiac muscle, they're a little further away, and then in smooth muscle, they're like way far away.
Okay, so it's proportional.
And in smooth muscle, you don't have these T tubules at all. So super interesting. There's kind of the cycle that the myosin head goes through.
Did
and
you
here's
guys,
what?
Did you guys talk about any disorders where this gets
That's with?
erroneous? Yeah.
No.
Okay.
We probably will. I'll ask about that.
So I was curious because, yeah, in other ones, there's ones where ATP irreversibly binds and it can't be hydrolyzed.
Oh, that's not great.
But I don't think that would actually, I guess if you bound ATP and it was non-hydrolyzable, then you'd be stuck relaxed, not contracted.
That's what this one's called. No, that's about speed. I'm pretty sure about that. There's another one in there, but I can't remember what it is. So, your skeletal muscle, you've got fast twitch and slow twitch muscles, and then the fast ones, you've got two different kinds. You've got the 2A and 2B. So, these are, muscle, fiber, diameter, or size, blood supply, mitochondria, color, really interesting, fatiguing, energy source. Okay, so your slow muscle fibers, those are ones that are non-fatiguing. So these are ones that are like literally always in use. So a good example of a slow muscle is your soleus, because you're literally using that all the time to like be standing, to walk.
Balance.
And it would be really bad if that just decided to just give out.
Take a rest.
Yeah, and so these non-fatiguing ones, so they've got small diameter, and they have a really extensive blood supply, and they have a high amount of mitochondria because you want that oxidative aerobic respiration.
Right.
They are very red, and then their energy source, this was oxidative glycolysis and respiration. So these are ones that are like literally being used basically all the time. These ones, medium, and then your type 2, really big diameter fibers. Blood supply, also medium. For the large ones, it's actually really low. For example of one of these ones is kind of like your biceps.
Okay.
They're for color, mitochondrial amount, also medium, and then these ones are low. And then fatiguing, medium, and then these fatigue really fast. Color, these are red, these ones are white. So something like your biceps, you can use it a lot really quickly, but then it fatigues super quickly because it doesn't have as extensive a blood supply,
Right.
high amount of mitochondria, fatigues really quickly, but then you rest, and then you can do it again. Energy source, so your 2A are also oxidative, aerobic, but these ones are non-oxidative. These ones are not oxidative. These are anaerobic. Yeah, anaerobic. And so the different types of energy supplies we have, so there's cytosolic ATP, there's creatine, phosphate, there is just regular glycolysis, and then each of these is anaerobic, and then we have, respiration. So you have enough cytosolic ATP to contract the muscle for about 5 seconds. Creatine phosphate, basically what it is so it allows the cell to turn ADP more easily into ATP with the creatine phosphate in there.
Right.
And that'll get you about 10 seconds. The glycolysis can get you about 60 seconds. And then this could get you like hours. So your slow muscle fibers will use way more respiration, and then your fast ones will use these ones. It's like, the really interesting thing is the kinds of muscle fibers you have is genetic. You can't change it.
Right.
So if you've got people that are like insane, like Olympic sprinters, super, super high amounts of these and those.
Yeah.
And then you've got like ultra marathon runners, tons of the slow fibers. And then even more interestingly enough is women have more slow than men do. One of the examples in class that we got given was Hunt doing like hanging against another girl, and he could. Yeah, it was just hanging.
I don't think Hunt would appreciate
hanging like a hang challenge versus like some other like sports girl would use in college or high school. I don't remember which one. And so he was able to make it like a minute or so, and then she's still hanging after like 15 minutes.
Yep.
Like,
can just hang all day.
yeah, it's insane.
But if it was pull-ups, then he'd probably.
Oh yeah, no, he destroyed her in pull-ups.
Yeah.
So yeah.
I find it very interesting. In boa constrictors, so they can actually regulate their mitochondria. It's crazy. So if they eat an antelope, for instance, then they can upregulate their mitochondria up to 70 times the resting rate just for 2 weeks to process all that meat, and then they can downregulate it to hardly anything even going on.
Just to keep going with such a small amount of food for a while.
Yeah, because they can survive 6 months to a year without a meal.
That's insane. Okay, did I miss anything?
I
I don't think I did.
Advantages and disadvantages.
Yeah, we talked about all that.
Did you go over types of smooth muscle?
No, that's question 2.
Oh, okay.
This is just question
okay.
1.
I was like, wait. sources of energy.
Yes, we're talking about muscle fibers, channels, calcium action potentials, sarcomeres.
Yeah, I think you hit it all.
Great.